Hello there, and welcome to Sleepy Curiosity channel. As you find your comfortable position, let your body begin to release the weight of the day. If this quiet journey is already beginning to slow your thoughts, consider subscribing now, so these stories can keep finding you night after night. Now, let the relaxation settle into your breath. Tonight we are going somewhere close. Not to a distant galaxy, not to a buried civilization, not to the far edge of recorded human history. We are going to the ocean, and we are going to spend time with an animal that, the more carefully you look at it, the harder it becomes to look away. There is something unusual about the way this particular animal sits in our understanding of the natural world. It does not fit comfortably into the categories we normally use when we think about intelligence, about awareness, about what it means to have a mind. And yet the more science looks at it, the more those categories seem to need expanding. Not discarding. Not replacing. Just expanding, to make room for something that arrived at remarkable places by a completely different road. The octopus has been alive, in one form or another, for hundreds of millions of years. It has survived every mass extinction the planet has thrown at the history of life. It has watched, from beneath the surface of ancient seas, as the first forests grew on dry land, as the first vertebrates crawled out of the water and took uncertain steps into air, as the dinosaurs rose and filled the world and then disappeared in geological silence. And through all of that, in one form or another, it continued. It adapted. It became something that no one who encounters it for the first time quite knows what to do with. That quality of not quite knowing what to do with it is worth paying attention to. It is not confusion exactly. It is more like the feeling of encountering something that is both familiar and deeply foreign at the same time, something that shares enough with us to be recognizable as a kind of mind, and different enough from us that the recognition keeps surprising us. The octopus produces that feeling reliably. In researchers who have studied it for decades. In aquarists who work with it every day. In anyone who has ever looked into its eye and found, unexpectedly, something looking back. Let us begin, then, at the very beginning. Not the beginning of the octopus specifically, but the beginning of the story that eventually produced it. There is a question worth starting with. A question that sounds almost philosophical the first time you hear it, and that turns out to be one of the most precisely biological questions you can ask. What do you and an octopus have in common? Go ahead and let that question rest for a moment. There is no hurry. The answer will arrive in its own time, the way most good answers do. If you have seen an octopus, even in a photograph or a documentary, the image that comes to mind is probably striking. Something soft and fluid and boneless, moving through water in ways that seem to defy the usual rules about how bodies work. 8 arms that appear to have a kind of independent life to them, each one exploring and curling and reaching in directions that the other 7 seem barely aware of. Eyes that are large and oddly expressive for an invertebrate, positioned on the sides of a head that does not look quite like a head. A body that changes color and texture as you watch, in a constant restless conversation with whatever surface it happens to be near. And if you picture yourself alongside that image, the contrast seems almost absolute. You have a spine. The octopus has none. You have a single centralized brain. The octopus has something far more distributed and strange. You are a mammal, warm-blooded, air-breathing, your body organized around a skeleton that gives it structure and shape. The octopus is almost entirely soft, living underwater, its body a kind of organized fluid that can take almost any shape it needs to. And yet, the answer to the question is more than you would think. Quite a lot, actually. More than 500 million years ago, in the shallow waters of an ocean that no longer exists, there lived a common ancestor shared between the octopus and you. Not a fish. Not something with a spine or a recognizable brain or complex eyes. Something far simpler. Something that, if you could see it today, you would probably not recognize as an animal at all. Likely something resembling a soft, flat worm, without a defined brain, without complex eyes, without any of the structures we now associate with intelligence or awareness or sophisticated life. Just alive. Moving slowly through ancient water. Responding to its environment in ways that were probably not much more complex than the responses of a modern single-celled organism, just implemented in a body that was slightly more elaborate. Eating. Avoiding things that might eat it. Reproducing. Existing without any apparent destination or purpose beyond the immediate facts of its existence. And from that single, unremarkable starting point, 2 lineages separated. 2 paths diverging across hundreds of millions of years of completely independent evolution, shaped by completely different environments, pressured by completely different predators and prey, arriving eventually at completely different bodies and completely different nervous systems and, just possibly, completely different but equally real forms of awareness. One path eventually led to fish, to amphibians, to reptiles, to mammals, to the explosion of primate diversity that eventually produced, among many other things, you, sitting or lying in this particular position, in this particular place, at this particular hour, with a mind that can contemplate its own origins. The other path led somewhere else entirely. That other path led to the octopus. This separation has an implication that biologists find genuinely remarkable, and that becomes more interesting the longer you sit with it and let it settle. If something like thinking exists inside an octopus, if there is something happening in that body that resembles awareness or problem-solving or curiosity or any of the things we associate with a mind that is genuinely engaged with the world around it, then it did not come from the same place as our thinking. It is not a simpler version of mammalian intelligence, a preliminary sketch that the vertebrate lineage later refined into something more complete. It is not an earlier step in the evolution of the mind as we know it, a stage that our own ancestors passed through on their way to becoming us. It is something that arose differently, shaped by completely different pressures, built from completely different materials, running on a completely different architecture, and arriving, after hundreds of millions of years of separate history, at results that are surprisingly, almost unsettlingly, similar to things we recognize in ourselves. Scientists call this convergent evolution, the phenomenon where two unrelated lineages independently arrive at similar solutions to similar problems, without any shared ancestry that could explain the similarity. Wings in birds and wings in bats are a classic example. Both evolved for flight, both use similar aerodynamic principles, but the specific bones and tissues involved are organized differently because they evolved from different ancestral structures. The eye is another example, and we will return to that one specifically when we talk about octopus vision. The octopus is, perhaps, the most striking example of convergent evolution in all of biology, because what has converged is not a physical structure like a wing or an eye, but something far harder to define and far more surprising to find twice. Something like a mind. Something like intelligence. Something like the capacity to learn, to remember, to solve problems, to be curious, to recognize individuals, to play. All of it appearing twice in the history of life on Earth, in lineages so distant from each other that they share no common ancestor with a nervous system worth speaking of. That is what makes the octopus so interesting to neuroscientists, so interesting to philosophers of mind, so interesting to anyone who wants to understand what intelligence actually is at its most fundamental level. It is not just an unusual animal. It is a natural experiment, one that the universe ran independently, with different starting materials and different constraints, arriving at a result that tells us something important about what is possible when you build a complex nervous system from scratch. The octopus belongs to a group called cephalopods, which also includes squids, cuttlefish, and the nautilus. The name means roughly head-footed, a reference to the way the arms emerge directly from the head region, without a separate neck or thorax intervening the way they do in vertebrates. It is a name that captures something real about the body plan, even if it sounds a little strange the first time you encounter it. This group has existed for at least 480 million years, a number that places it far beyond the emergence of any primate, far beyond the grasslands of the African savanna, far beyond the Alps, far beyond almost everything we think of as ancient when we are being generous with that word. When the lineage that would eventually become whales first began moving toward the water, cephalopods were already ancient. They had already been dominant predators of the ocean for longer than most living groups of animals had existed at all. It is worth trying to feel the weight of that number, even knowing that numbers this large resist being felt directly. 480 million years ago, the continents were arranged differently. The atmosphere had a different composition. The sun rose and set over a world that would be almost completely unrecognizable to any observer familiar only with the modern Earth. And yet in the ocean of that world, cephalopods were already there. Already swimming. Already hunting. Already finding ways to survive in environments that would eventually swallow countless other lineages without a trace. What is remarkable about this history is not just its length but its endurance, the way the group passed through conditions that ended everything else around it and kept going. 5 mass extinctions, each one catastrophic enough to eliminate a substantial fraction of all life on Earth. The end Ordovician extinction, approximately 440 million years ago. The late Devonian extinction. The Permian-Triassic extinction, the largest of all, which eliminated perhaps 90% of all species on Earth, an event so catastrophic that geologists can identify it in rock formations around the world as a distinct boundary layer, a moment where the fossil record simply empties out and then, slowly, haltingly, begins to refill. Then the Triassic-Jurassic extinction. And finally the Cretaceous-Paleogene extinction, the one that ended the non-avian dinosaurs 66 million years ago. The cephalopod survived all of them. Not without losses, not unchanged, but the group passed through each of those boundaries in geological time and continued diversifying on the other side. The octopus belongs to a lineage of extraordinary survivors, animals shaped by pressures that most creatures alive today have never encountered, carrying in their biology the marks of 5 separate near-total resets of life on Earth. Within that ancient lineage, the modern octopus is relatively recent. It inherited a formula tested across hundreds of millions of years and refined it in a very specific direction: less structure, more flexibility, more neural complexity, more behavioral sophistication. As if evolution had taken an already successful platform and decided to push certain features toward their limits, to see how far they could go. One of the reasons the fossil record of octopuses is so sparse is that the animal practically never leaves a trace when it dies. The body is almost entirely soft. No bones, no shell, no mineralized structures that resist the process of fossilization. In the vast majority of cases, an octopus that dies decomposes completely without leaving anything behind in the rock, no impression, no outline, no chemical signature robust enough to survive millions of years of geological pressure and transformation. The octopus passes through the world, for most of its evolutionary history, without leaving the kind of record that allows us to reconstruct that history with confidence. Only a handful of well-preserved octopus fossils exist anywhere in the world, and the most famous of them, found in limestone deposits in Lebanon, is approximately 95 million years old. What struck the paleontologists who studied it was how familiar it looked. The general structure of the body, the arrangement of the arms, even evidence of an ink sac, already present. In roughly 100 million years, the basic form of the animal had changed very little. This puts the octopus in an interesting category, not quite a living fossil in the classic sense, because internally, especially in the development of the nervous system, the animal kept evolving and complexifying in ways that living fossils by definition do not. But its outward architecture found an equilibrium early and stayed there, because the architecture worked. The body plan solved the problems it needed to solve with enough efficiency that the large-scale selection pressure to dramatically change it was low. What kept changing, beneath that stable exterior, was the neural complexity, the behavioral repertoire, the capacity to learn and remember and adapt. The external form stabilized while the internal sophistication continued to grow. It is an unusual combination, the stability of an ancient body plan housing a nervous system that kept on developing long after the body itself stopped changing much. Like a building whose facade has remained the same for a century while the interior has been completely renovated, floor by floor, room by room, until the inside bears almost no resemblance to what it was when the exterior was new. The ancestors of the octopus had shells. External, protective shells, coiled or straight, made of calcium carbonate deposited in precise geometric layers by the mantle tissue, the same kind of structure found in snails and clams and oysters, which are distant relatives in the great molluscan lineage. For most of cephalopod history, the shell was a central feature of the group's success, providing protection in oceans full of predators, and in some lineages providing buoyancy control through gas-filled chambers that the animal could fill or empty to rise and sink in the water column. The nautilus is the living reminder of this ancestral condition. Coiled in its beautiful spiral shell, navigating the ocean by adjusting the gas in its chambered interior, the nautilus exists essentially unchanged for 500 million years, carrying its house with it the way the lineage always did, the way every cephalopod once did, in an ocean that no longer contains most of the other animals that shared it then. The nautilus is a kind of geological survivor, a living snapshot of a world that otherwise exists only in rock. But at some point in the history of the cephalopod lineage, something changed. Some lineages began reducing the shell, internalizing it, shrinking it, moving it inside the mantle where it provided some structural support without the full protective investment of an external armored casing. Squids have what remains of that ancestral shell as a thin, transparent internal sliver called the gladius, a structure so reduced and modified from its origins that you would not recognize it as related to the nautilus shell without the evolutionary history to connect them. Cuttlefish have the cuttlebone, that chalky white structure sometimes sold as a calcium supplement for pet birds, a denser and more substantial remnant than the squid's gladius but still a far cry from the robust external shell of the nautilus. The octopus went further than any of them. It eliminated the structure entirely. No gladius, no cuttlebone, no internal remnant of any kind, no chemical trace in the tissue that might suggest a reduced structure was ever there and then disappeared. The complete elimination of any trace of the ancestral shell is unique to octopuses within the entire cephalopod group, a distinction that seems minor when you first hear it, but that turns out to have enormous consequences for everything that follows. This was, from any reasonable engineering perspective, an enormous gamble. The shell was protection. It was the reason cephalopods had been so successful for so long. Without it, the animal was exposed in ways it had never been before, soft tissue available to any tooth or beak or claw that could reach it, no layer of calcium carbonate standing between the living body and the world's willingness to consume it. A predator that could not pierce the shell of a nautilus could very easily injure an octopus. The vulnerability was real, immediate, and permanent. But the counterpart to that vulnerability was transformative in ways that are difficult to fully appreciate without thinking carefully about what a shell actually does to the body that carries it. A shell constrains. It defines a shape, and that shape is the animal's shape for as long as the shell lasts, and the shell in most mollusks lasts for life. The animal grows, slowly filling the spiral or expanding the opening, but the fundamental geometry of the structure is fixed from the moment it begins to form. You cannot fold a shell. You cannot compress it. You cannot flow it through an opening narrower than its diameter. A body without a shell is a body that has been freed from that constraint entirely. A body that can be reshaped into almost anything, that can squeeze through any opening, flow into any crevice, wrap around any shape, compress itself against any surface with a degree of conformity that a shelled animal can never achieve. The mobility and flexibility that came from this loss turned out to be worth far more than the protection that was sacrificed. Not because protection doesn't matter, but because flexibility, in the right ecological context, can substitute for protection in ways that are just as effective and considerably more versatile. Many researchers believe that this loss of the shell, may have been one of the primary drivers of octopus intelligence. Not a consequence of intelligence, not something that evolved alongside it, but a cause, a pressure that made intelligence necessary in a way it had not been before. The reasoning is direct and fairly simple to follow. A soft-bodied animal living in an ocean full of predators needs alternative survival strategies if it is going to survive at all. Sophisticated camouflage, so that predators cannot find it. Rapid escape mechanisms, so that it can flee when camouflage fails. Recognition of specific threats, so that it can distinguish between a dangerous predator and a harmless fish and respond appropriately to each. Memory of safe locations, escape routes, productive hunting grounds, places where predators tend to patrol. Problem-solving capacity for situations that could not be anticipated in advance. All of these things become enormously valuable when physical protection is removed from the equation, when there is no shell to fall back on, when the body itself is the only thing standing between the animal and everything that wants to eat it. There is a broader hypothesis in evolutionary biology suggesting that environments that demand behavioral flexibility, where threats vary unpredictably, where fixed instinctive responses are not sufficient because the situations keep changing, where the animal must figure things out rather than simply executing a pre-programmed response, tend to favor the development of more complex nervous systems. A brain that can learn and adapt is worth more than a brain that can only react when the environment is genuinely unpredictable. The investment in neural complexity pays off only when flexibility actually matters. The octopus, in losing its shell, placed itself involuntarily into exactly that kind of environment. Each generation that survived did so partly by being a little more capable of learning, of recognizing patterns, of improvising solutions to novel problems. Each generation that was slightly better at this survived slightly more reliably than each generation that was slightly worse at it. Across millions of generations, across a time span that is genuinely difficult to conceptualize, that pressure accumulated. It built, slowly and without any destination in mind, toward something that no one looking at a soft worm ancestor 500 million years ago could have predicted. It built toward the octopus as we find it today, an animal that demonstrates capacities that we normally associate exclusively with vertebrates, that appears in many experimental contexts to be doing something that looks very much like thinking, that continues to surprise researchers who thought they knew what to expect from an invertebrate. The shell was lost, and something else was gained in its place. Something that, from a certain angle, looks like a much better deal. The complete genome of the octopus was sequenced for the first time in 2015 by an international team of researchers, and the results that emerged surprised even scientists who had been studying cephalopods for decades. The genome is large, comparable in total size to the human genome, containing approximately 2.7 billion base pairs and approximately 33,000 protein-coding genes. That last number is higher than our own. The human genome contains somewhere around 20 to 25,000 protein-coding genes, depending on how you count. The octopus has more. This is surprising in itself, because we tend to assume, in a vague and not entirely defensible way, that more complex or more intelligent animals should have more genes, as if genes were the currency of biological complexity. But genomic complexity and organismal complexity do not map onto each other that simply. What the gene count tells you is not how sophisticated the animal is, it tells you something about the history of the genome itself, the duplications and expansions and losses that have accumulated over evolutionary time. What is more informative than the total count is what those genes do. And a disproportionate number of the octopus genes, far more than you would expect by chance, are associated with the development and function of the nervous system. The genome reflects, at a molecular level, the same investment in neural complexity that we see expressed in the animal's behavior. Among the most striking findings was a greatly expanded family of genes called protocadherins, proteins involved in forming and organizing the connections between neurons, the synapses through which nerve cells communicate with each other. In vertebrates, protocadherins play a fundamental role in building the architecture of the brain. They help neurons find the right partners, establish the right connections, organize themselves into the circuits that eventually give rise to memory, learning, and cognition. Finding them in octopuses, which evolved completely independently of any vertebrate, was one of the clearest molecular demonstrations of convergent evolution ever documented. Similar genetic tools, arriving through completely separate evolutionary paths, refined by completely separate selection pressures, to solve similar problems in the construction of a complex nervous system. The researchers also found evidence of an unusual form of genetic regulation called RNA editing, which is extraordinarily rare in animals but occurs at very high rates in octopus neurons. To understand why this is interesting, it helps to know a little about how genetic information normally works. The standard model is that DNA encodes the instructions, RNA reads those instructions and carries them to the cellular machinery that builds proteins, and the proteins do the actual work of the cell. This is normally a fairly faithful copying process. The RNA message that leaves the nucleus reflects the DNA sequence with high accuracy. RNA editing is an exception to this. It is a process by which the cell can alter the RNA message after it has been read from the DNA, changing specific nucleotide bases in the message and thereby changing what protein gets built, without changing the DNA itself. It is like being able to modify the printed recipe after it leaves the cookbook but before the cook reads it. The result is a layer of flexibility in gene expression that goes beyond what the DNA sequence alone would predict. In most animals, this kind of editing is rare and tightly controlled. In octopus neurons, it happens constantly, at an extraordinary rate, affecting a significant fraction of the proteins that the nervous system produces. The implications of this for the flexibility and adaptability of the octopus nervous system are still being worked out, but the prevalence of the process in neural tissue specifically suggests that it contributes something important to how the octopus brain functions. It may allow the nervous system to fine-tune its own operations in response to experience in ways that are faster and more flexible than traditional genetic mechanisms would permit. The octopus has no bone anywhere in its body, with one exception. This is worth sitting with for a moment before continuing, because it is the kind of fact that sounds simple and turns out to have far-reaching consequences once you start following where it leads. No bone. No skeleton. No internal framework of any kind except for the single exception we are about to describe. In a body that can range from a few centimeters to several meters in span, depending on species, in a body containing all the organs needed for complex life, 3 hearts, 2 kidneys, a digestive system, a complex nervous system, reproductive organs, a sophisticated visual system, there is no rigid internal structure providing support or leverage. The body holds its shape and generates its movements entirely through the coordinated contraction of muscle, supplemented by the hydraulic pressure of fluids within the tissues. The single exception is the beak. Located at the center of the tentacles on the underside of the animal, at the point where all 8 arms converge on the mouth, the beak is made of chitin, the same hard biological polymer that forms the exoskeleton of insects and crustaceans. It is hinged in 2 sections that work like scissors, with the upper section fitting inside the lower section the way a parrot's beak works, and it is surprisingly powerful for its size. Capable of cutting through the shells of crabs, puncturing the hard casings of mollusks, severing the scales of fish, it applies considerable force with precision, in a way that no other structure in the octopus's body can replicate. But the beak has a second function that is less about what it can do and more about what it is. Because it is the only rigid, non-compressible structure in the entire animal, it sets a physical limit. The beak defines the minimum diameter of any opening through which the octopus can pass its body. Everything else, every cubic centimeter of muscle and organ and nervous tissue and fluid, is compressible. It can be squeezed, reshaped, redistributed, flattened, folded. But the beak cannot. If the beak fits through an opening, the rest of the body can follow. If the beak does not fit, the octopus cannot pass, no matter how soft and compressible everything else is. The practical consequences of this principle are well documented in aquariums and research facilities around the world, and they are consistently astonishing to people who encounter them for the first time. Octopuses escape from tanks considered secure, identifying gaps and tube connections and overflow channels that human engineers had not thought to seal, because human engineers did not anticipate an animal that can compress its body to the diameter of its own beak. They appear in neighboring tanks, having traveled through plumbing that should have been too narrow, hunt the fish or invertebrates they find there, and sometimes return to their own tank before the morning shift arrives and notices anything unusual. In more than a few documented cases, the staff only discovered what was happening after installing cameras and reviewing the overnight footage. The beak, in this context, is not just a feeding tool. It is the parameter that defines the physical limits of an animal that, in every other sense, seems to have almost none. The body of the octopus is composed almost entirely of a very specific kind of muscle, what biologists call hydrostatic muscle. This is the same kind of muscle found in the human tongue, in the elephant's trunk, in the tentacles of sea anemones, structures that generate movement not by contracting against a rigid bone, the way virtually all vertebrate muscles do, but by contracting in different directions simultaneously, using the incompressibility of fluid within the tissue as internal structural support. To understand how this works, it helps to think about what a conventional skeletal muscle actually does when it contracts. It shortens. That's all. The muscle pulls its two attachment points closer together, and the bones those attachment points are attached to rotate around the joint between them, producing the lever action movement that gives vertebrate limbs their range of motion. The muscle can only pull, never push, which is why vertebrate muscles come in pairs that work against each other, one to flex the joint, one to extend it. And the range of motion is limited by the geometry of the skeleton, by the angles that the joints allow, by the length of the bones involved. Hydrostatic muscle works on a completely different principle. When muscle fibers contract in one direction in a hydrostatic structure, the incompressible fluid within the tissue cannot go anywhere, so it redistributes into other directions. If you squeeze a water-filled balloon from the sides, it elongates. If you squeeze it from the ends, it widens. Hydrostatic muscles exploit this principle to produce movement in multiple directions through selective contraction, without any rigid elements at all. Contracting the longitudinal fibers shortens the arm and thickens it. Contracting the transverse fibers elongates the arm and thins it. Contracting oblique fibers in different combinations produces twisting and bending in any direction. The result is a system with, effectively, infinite degrees of freedom at every point along the arm's length. An octopus arm can stiffen into a rod, go completely limp, shorten to half its length, extend to full stretch, curl into a spiral, flatten into a ribbon, or bend in any direction at any point along its entire length, all in the same second, all in response to neural signals that the arm's own nervous system coordinates largely without input from the central brain. The range of possible shapes and movements is, as far as mechanics is concerned, unlimited. This is not hyperbole. The mathematics of hydrostatic structures confirms it. There is no position the arm cannot assume, no curvature it cannot achieve, no configuration that the muscle arrangement cannot produce. For engineers and roboticists working in the field called soft robotics, the field that seeks to build machines with flexible, adaptable bodies capable of operating in unstructured environments where rigid robots struggle, the octopus arm is the primary biological reference, the existence proof that this kind of structure can work, can be controlled, can produce useful and precise behavior. The challenge they keep encountering is not finding the right materials. Materials that approximate the mechanical properties of hydrostatic muscle exist and are being refined. The challenge is control. How do you coordinate a structure with infinite degrees of freedom to produce useful, directed behavior? How does a nervous system solve that problem reliably, in real time, in a body that is moving through an unpredictable environment? That question is still not fully answered, either in biology or in engineering. Each of the 8 arms of an octopus is covered in rows of suckers, and these suckers are considerably more sophisticated than they appear to casual observation. Each one is a complete sensory unit in its own right, not just a mechanical gripping device but a multimodal sensor that combines several kinds of information in a single structure. On the mechanical side, each sucker contains receptors sensitive to pressure and to texture, to the shape and roughness and compliance of whatever surface it contacts. These mechanoreceptors can distinguish surfaces that differ in roughness by only a few micrometers, smaller than the diameter of a human hair, comparable to the resolution of the human fingertip, which is considered one of the most sensitive touch surfaces in the biological world. When an octopus arm moves across an object, it is building a detailed tactile map of that object's surface in real time. On the chemical side, each sucker also contains receptors that function essentially as taste receptors, chemoreceptors capable of binding to molecular compounds present in the environment and generating signals that tell the nervous system what those compounds are. An octopus touching an object is not only feeling its texture and shape. It is simultaneously tasting its chemical composition, detecting the specific molecules that make up its surface, distinguishing prey from rock, from plastic, from shell with a chemical specificity that goes far beyond anything we can do with our own sense of touch. These two sensory modalities, touch and taste, are combined in a single structure at every one of the thousands of suckers distributed across 8 arms. A medium-sized octopus may have between 1,600 and 2,400 suckers, each one sending continuous signals while the arm is in contact with the environment. The total volume of sensory information being generated at any given moment during active exploration is enormous. And here is where the architecture of the octopus nervous system becomes directly relevant. Most of that information is processed locally, in the ganglia, the local nerve clusters, distributed along the length of each arm. It does not all travel to the central brain. It does not need to. The arms can process much of the information they receive at the point of reception, making local decisions about what to grasp, what to taste further, what to release, without waiting for the central brain to weigh in. Only a fraction of the information collected by the arms reaches the central brain at all, a summary, a compressed digest of what the arms are experiencing, rather than the full raw feed. But the most significant feature of the octopus nervous system is not any individual structure. It is the distribution of the whole. An octopus has a central brain, a compact mass of neural tissue located between the eyes, wrapping around the esophagus, containing approximately 50 million neurons. This is the part of the nervous system that processes visual information from the eyes, coordinates overall behavioral goals, integrates information from across the body, and maintains what we might loosely call the animal's overall sense of what is happening and what to do about it. But that central brain accounts for only about 1/3 of the animal's total neurons. The other 2/3 are distributed, roughly 50 million neurons per arm, organized into ganglion clusters that run along the length of each arm like a series of local processing centers. These ganglia do not merely relay information to the central brain. They process it. They make decisions. They initiate and coordinate movements. They can solve problems and respond to stimuli entirely on their own, without any signal from the central brain at all. The central brain sends general commands, something like explore this area or capture that prey or move toward the light. And the arms carry out the specifics of those commands independently, using their own neural resources to figure out the details. The central brain does not micromanage. It delegates. And the arms are genuinely capable of executing complex behavior on the basis of that delegation, without constant central supervision. The most direct and most striking evidence of this autonomy comes from experiments with amputated arms. When an octopus arm is surgically removed and maintained in physiological conditions, oxygenated saltwater at the right temperature and pH, the basic conditions needed to keep the tissue alive and functional, it continues responding to stimuli for up to an hour after separation from the body. This alone is remarkable. An arm, detached from any central brain, continues to respond to the world. But the responses are not random twitching. They are organized behavior. When the detached arm detects food, through the chemical receptors in its suckers, it attempts to grasp the food and carry it toward where the mouth would have been, in the direction that would have been correct if the arm were still attached to a body with a mouth at its center. The arm has no mouth to deliver food to. It has no body at all. But it executes the capture and deliver sequence anyway, because that sequence is encoded in the arm's own nervous system, not in the central brain, and the arm's nervous system is still functioning. The arm behaves as if it still knows what it is supposed to be doing. It knows, in some sense, that the arm's ganglia implement without any central supervision, that when you detect food you grasp it and bring it toward the mouth. And it does this even without a mouth, even without a body, even without any connection to any central processing system that could be directing it. The behavior is local knowledge, embedded in the arm's own neural tissue, executing without oversight because it never required oversight in the first place. This creates a question that researchers do not avoid asking, even when the question gets uncomfortable. A question that sounds philosophical but has a real neurological basis. When we say that an octopus thinks, where, exactly, is that thinking happening? In the central brain? In the arms? In some ongoing conversation between the two that doesn't clearly belong to either? Is there a unified subject of experience somewhere in this distributed system, a single point that aggregates everything and is, in some meaningful sense, the octopus? Or is the octopus something more like a committee, 9 processing centers, each with its own information and its own local intelligence, coordinating loosely toward shared behavioral goals without any one center being fully in charge? These are not questions with easy answers. And part of what makes the octopus so interesting to neuroscientists and philosophers alike is precisely the absence of easy answers. The animal forces the question of what it means to have a unified mind, to have a single subject of experience, to be an individual in the way we normally mean when we use that word. Because in an octopus, the boundaries of individuality are drawn at a different scale than they are in any vertebrate we know. There is a structural detail of the octopus central brain that is worth pausing on because it adds another layer to this picture. The brain wraps around the esophagus. The tube through which food passes from the mouth to the stomach runs directly through the middle of the central brain, so that every time the octopus swallows, the food passes literally through the brain on its way down. This means the octopus cannot swallow anything too large without risk of compressing its own nervous tissue. For an animal with a beak capable of cutting through crab shells, the size of prey that can actually be swallowed is limited not by the strength of the feeding structures but by the size of the hole at the center of the brain. This is the kind of anatomical constraint that evolution works around but rarely eliminates entirely. It is a reminder that the octopus nervous system, for all its sophistication, was not designed from scratch by an engineer with a blank slate. It evolved incrementally, with each modification building on what already existed, and some of what already existed was not ideally positioned for the uses it was eventually put to. The brain wrapped around the esophagus because the esophagus was there first, and the brain grew around it. And now both are there, in that slightly awkward arrangement, doing the best they can with the geometry they inherited. Some researchers who have studied the relationship between the central brain and the arm ganglia in detail describe a situation where the central brain appears to have only partial awareness of what its own arms are doing in real time. In experiments where an octopus arm was guided to touch another part of the animal's own body, the octopus frequently reacted as if being touched by an external object, flinching, reorienting, attempting to pull away from the contact. The central brain, it seems, did not have precise moment-to-moment information about the position of its own limbs. It did not know, in the way that you know where your hand is even with your eyes closed, exactly where each arm was and what each arm was doing at every instant. If that is accurate, and the experimental evidence suggests it is, then the octopus lives in a situation that has no close parallel in vertebrate neurology. Part of its own body operates with a degree of independence that the center does not fully monitor. The central brain sets goals and receives summaries, but the details of execution happen elsewhere, in neural tissue that is doing its own processing without reporting back everything it knows. Whatever the subjective experience of that is, if there is anything like subjective experience in an octopus at all, it must be structured in a way that is fundamentally different from anything a vertebrate nervous system produces. Not necessarily less rich. Not necessarily less aware. Just different. Organized around different principles. Drawing boundaries between self and world in ways that our own experience does not prepare us to imagine. And that difference is, in its own way, one of the most interesting things about the octopus. Not just as a biological curiosity, but as a window onto the question of what a mind can be, how many different forms awareness might take, How many different architectures might support something like experience in a universe that has been running evolutionary experiments for 500 million years and has not yet finished? The skin of the octopus is probably the most sophisticated camouflage system found in any animal on Earth. That is not a casual claim. It is the considered judgment of researchers who have spent careers studying animal coloration across hundreds of species, and who return consistently to the octopus as the benchmark against which every other system is measured and found, in some significant respect, simpler. To understand why, it helps to start with what the skin actually has to accomplish. The octopus is soft, slow relative to many of the fish that prey on it, and lives in environments that are visually complex and variable, reef systems with elaborate geometry and shifting light, sandy floors that seem uniform at a glance but vary in color and texture in ways that matter to a precise camouflage system, rocky substrates with irregular surfaces and unpredictable shadow patterns. The animal needs to match this environment precisely, in real time, from any angle of observation, while potentially moving through it, and it needs to do this while being, as we will discuss in a moment, almost certainly colorblind. The skin accomplishes this through 3 distinct layers of specialized cells, each doing something different, all working simultaneously. The outermost layer contains the chromatophores. These are not pigment cells in the passive sense, the way the melanocytes in human skin produce pigment that simply sits there and absorbs certain wavelengths of light. Chromatophores are active mechanical devices. Each one is a small elastic sac filled with pigment, surrounded by a ring of tiny muscles, called radial muscles, because they radiate outward from the sac like the spokes of a wheel. When these muscles contract, they pull the walls of the sac outward, stretching it flat and spreading the pigment over a larger area, making the color visible. When the muscles relax, the elastic sac snaps back to a small sphere, concentrating the pigment in a tiny point that is effectively invisible against the skin. The color does not change. The size of the pigment area changes, from a flat disk a fraction of a millimeter across to a point too small to see, in milliseconds. Each chromatophore is controlled individually by its own dedicated motor neurons, neural connections that run directly from the nervous system to the muscles of that specific chromatophore, allowing independent control of each unit. In some species, the density of chromatophores reaches 200 per square millimeter of skin. That is an enormous number. It means the skin has, effectively, a spatial resolution comparable to a high-quality printed image, with each pixel independently controllable at the speed of neural transmission. Changes do not happen gradually, through diffusion of pigment or slow chemical reactions. They happen at neural speed, because they are driven directly by neural signals. A complete change of color pattern can occur across the entire body in less than a second. Beneath the chromatophore layer are the iridophores. These cells work on a completely different optical principle. They contain no pigment at all. Instead, They contained stacks of thin protein plates, each plate only a few hundred nanometers thick, with gaps of a similar dimension between them. At these scales, light waves interact with the structure itself, reflecting off successive surfaces and interfering with each other in ways that produce color, the same phenomenon responsible for the colors of oil slicks, soap bubbles, and butterfly wings. The color produced by structural interference is different from pigment color in important ways. It can be iridescent, changing with viewing angle. It can produce metallic sheens and brilliant blues and greens that would be difficult or impossible to achieve with pigment alone. And in some species of octopus, the iridophores can be actively controlled by the nervous system, changing their reflective properties in response to neural signals in ways that add a dynamic dimension to the structural color that is still being characterized by researchers. The deepest layer contains the leucophores, cells that scatter light broadly, reflecting all wavelengths diffusely and producing a bright, non-directional white. These function as a neutral background, a base layer over which the chromatophores and iridophores operate, providing the reflective foundation that makes the upper layer's effects visible. The interaction between these three layers creates an optical system of extraordinary versatility. A chromatophore expanded over an iridophore filters the light reaching the iridophore, modifying the structural color that results. A chromatophore contracted over an iridophore allows the iridescent effect to shine through directly. Different combinations produce different results at every point in the skin, and all of these combinations can change simultaneously and independently across the entire body surface, at neural speed, in response to visual information from the eyes and possibly from the skin itself. No material designed and built by human engineering comes close to replicating this system. Not in speed. Not in spatial resolution. Not in the range of effects achievable. Not in the ability to combine chromatic, structural, and diffuse reflective properties in a single dynamically controllable surface. Engineers working on adaptive camouflage, on next-generation display technologies, on materials that can change their optical properties on command, study the octopus skin as a design goal, something to aspire toward, a proof that the physics permits what they are trying to achieve, even if the biological implementation remains beyond their current ability to replicate. Beyond color, the skin controls its own physical texture, and this is a dimension of the octopus's camouflage system that is easy to overlook, but that turns out to be at least as important as color for effective concealment. Scattered across the skin are structures called papillae, columns of muscle tissue that can be raised from the skin's surface like bumps, ridges, or branching protrusions, and then retracted back to leave the skin completely smooth. They come in different sizes, small papillae that create a fine granular texture, larger ones that produce substantial ridges and peaks, and in some species very large ones that can protrude several centimeters from the body surface, creating dramatic three-dimensional structures that break the smooth outline of the body in ways that color alone cannot achieve. The significance of texture to effective camouflage is something that visual ecologists have studied carefully. Color matching is important, but an observer familiar with the body shape of an octopus can potentially identify it even against a well-matched color background, because the outline of the body remains recognizable. An oval blob of a specific size and proportion is still an octopus even if its color matches the surrounding rock. But if that outline is broken by protrusions that echo the irregular surface of algae, or coral, or a sponge, or a rock encrusted with biological growth, the shape becomes ambiguous, harder to resolve into the familiar form of a predator or a prey item. The texture interrupts the gestalt recognition process that many predators rely on. Octopuses in environments with branching algae can erect papillae that produce a surface resembling the fronds of those algae, complete with irregular branching protrusions of different heights. Octopuses on coarse sandy or rocky substrates can produce a low, granular texture that matches the roughness of the ground. Octopuses resting in smooth-walled crevices can retract all papillae completely, presenting an unbroken smooth surface that matches their surroundings. And the transition between these states, between rough and smooth, between high contrast texture and flat uniformity, can happen in a fraction of a second, driven by the same neural speed that controls the chromatophores. The combination of real-time color change and real-time texture change, each operating independently and simultaneously over the entire body surface, produces a camouflage system whose output space is effectively unlimited. The animal can match not just the average color of its background but its spatial structure, its contrast pattern, its surface geometry, and the way light interacts with it, all at once, all in real time, adjusting continuously as the animal moves or as the light changes. Researchers who have studied octopus camouflage systematically have identified something interesting about how the system is organized. Rather than producing an infinite variety of fully customized patterns, the octopus appears to use a relatively small number of pattern categories, what researchers describe as uniform, mottled, and disruptive, and to adjust the specific parameters within each category to match the immediate environment. A uniform pattern is used on backgrounds that are relatively homogeneous in color and texture: flat sand, smooth rock, a single color substrate. The animal becomes approximately the same color and texture as the background, without internal contrast or pattern. A mottled pattern introduces irregular patches of lighter and darker areas, matching backgrounds that have moderate local variation: gravel, algae-covered rock, substrates with scattered elements. A disruptive pattern uses high-contrast patches with abrupt edges, deliberately breaking up the body outline in ways that make it harder for an observer to resolve the whole into a recognizable shape, particularly effective on high-contrast backgrounds like reefs with sharp light-dark boundaries. This categorical structure simplifies the control problem considerably. Instead of computing a fully novel response to every new background, the system classifies the background into one of a small number of categories and then adjusts parameters within that category, the specific color, the specific contrast level, the specific texture, the specific scale of the pattern. The classification is fast because the categories are broad. The parameter adjustment is flexible because the underlying cellular system allows continuous variation within each category. The result is a system that is both fast and flexible, achieving effective camouflage quickly because the basic structure of the response is pre-organized, but adapting to specific conditions because the details can be tuned. The resolution of this system is remarkable enough to produce different patterns on different parts of the body simultaneously. When half of an octopus rests on sand and the other half rests on coral, the sand half and the coral half can produce different camouflage patterns at the same time, with a transition boundary that follows the edge between the two substrates. The skin is not receiving a single global instruction from the central brain. There must be a spatial mapping between what the visual system sees and which regions of the skin are supposed to respond to what, and that mapping must have a resolution fine enough to produce clean transitions at the boundary between two different backgrounds. How this spatial mapping works at the neural level is an active area of research. But its existence implies a kind of topographic organization in the system, a preserved correspondence between visual space as seen by the eyes and skin space as controlled by the nervous system, so that the command to match a particular region of the visual field is delivered to the corresponding region of the skin. This is the kind of spatial organization that we see in the visual cortices of vertebrates, where neighboring neurons tend to process information from neighboring regions of the visual field. Finding something analogous in the octopus camouflage system would be another example of convergent neural organization, the same solution to the same spatial correspondence problem, arrived at independently in a completely different nervous system. Now, there is one more thing about the octopus's relationship to color that needs to be addressed, because it is one of the most persistent puzzles in the biology of the group, and one of the most counterintuitive facts about an animal that seems otherwise so visually sophisticated. The octopus is almost certainly colorblind. Its retina contains only one type of photoreceptor, one variety of the light-sensitive cells that line the back of the eye and generate the signals that the visual system interprets as color. Color vision, as we understand it, requires at least two types of photoreceptors with different spectral sensitivities, so that the nervous system can compare the relative outputs of different receptor types and infer the wavelength of the incoming light. We have three types of photoreceptors, sensitive to roughly red, green, and blue wavelengths, and our color perception emerges from the brain's comparison of signals from all three. Animals with four types of photoreceptors, some birds, for example, perceive colors we cannot even imagine. The octopus has one type, a single spectral sensitivity. No comparison possible. In the conventional sense of the word, it cannot distinguish colors. And yet it produces color patterns that match its environment with a precision that is hard to explain without assuming it knows something about the colors it is creating. Octopuses in bright tropical reefs with complex, multicolored backgrounds produce complex multicolored camouflage. Octopuses on monochromatic sand produce monochromatic patterns. The match is not perfect, no camouflage system achieves perfection, but it is consistently and significantly better than chance. Better than an animal that is truly indifferent to color could produce. This paradox resisted explanation for a long time. And then a hypothesis emerged that is, if correct, one of the more elegant solutions to a biological puzzle that the field has seen in recent years. The octopus pupil is unusual in shape. It is not round, like the pupil of a human eye. It is elongated and complex, often described as W-shaped or crescent-shaped, a horizontal slit with irregular edges that is quite different from the simple expanding and contracting circle of most vertebrate eyes. This shape is not merely decorative. It has optical consequences. When a lens has a wide aperture, as the octopus's large eye does, and the incoming light passes through an aperture of this unusual shape, different wavelengths of light focus at slightly different points on the retina. This is chromatic aberration, an effect that camera designers work hard to eliminate because it blurs images, but that might, under the right conditions, carry information about the wavelength of incoming light that a single receptor system could use to make inferences about color. The W-shaped pupil might allow just enough chromatic aberration, distributed in a spatially controlled way across the retina, to give the octopus functional information about color even with a single photoreceptor type. This hypothesis is not fully proven. Testing it rigorously is technically challenging. You cannot simply ask the octopus what color it sees. But the behavioral evidence for color matching and the optical evidence for the chromatic aberration effect are both consistent with the hypothesis. And the elegance of the solution, using the very defect that most optical systems are designed to eliminate as a source of useful information, is the kind of thing that evolution produces with a frequency that never stops being surprising. The octopus eye, regardless of what it does with color, is worth examining in more detail because it is one of the clearest examples of convergent evolution in all of biology, and because the specific way in which it converged with the vertebrate eye, and the specific way in which it did not, tells us something interesting about how evolution works. The vertebrate eye and the cephalopod eye arrived at almost identical solutions to the problem of building a camera-style eye. Both have a cornea at the front to refract incoming light. Both have a lens to focus it. Both have a chamber of clear fluid behind the lens to maintain the eye's shape. Both have a retina at the back, covered in photoreceptors that convert light into neural signals. Both have an iris that can change the size of the opening to control the amount of light entering. From the outside, from almost any functional perspective, these two eyes are doing the same thing and doing it in the same way. But they arrived at this common solution from opposite directions, in one specific respect that has a real functional consequence. In the vertebrate retina, the photoreceptors are installed backward, with their light-sensitive tips pointing away from the incoming light, toward the back of the eye, so that light must pass through multiple layers of supporting cells before it reaches the sensitive surface. This backward installation creates a location where the optic nerve must gather all its connections and exit the eye by passing through the retinal surface, because there is no room for it on the illuminated side. At that location, there are no photoreceptors. The optic nerve takes up the space. That location is the blind spot, the small region of the visual field where our retinas are literally insensitive to light, and where the brain fills in the gap using information from surrounding regions, so seamlessly that we never notice it under normal circumstances. The octopus retina installs its photoreceptors the right way around, with the light-sensitive tips pointing toward the incoming light, the way any optical engineer would install them if starting from scratch. The optic nerve connects to the back of the retina, on the side away from the light, and exits the eye there without needing to pass through the photoreceptor layer. There is no blind spot. The octopus retina is, in this specific respect, better designed than the vertebrate retina. This difference exists not because evolution is smarter in octopuses than in vertebrates, but because the two eyes evolved from different ancestral structures. The vertebrate eye evolved from a skin-based photoreceptor in which the cells happen to be oriented with their sensitive ends pointing inward, away from the light. By the time the eye became complex enough for the orientation to matter, too much had been built on that ancestral foundation to simply reverse it. The backward orientation became locked in, a historical accident preserved by path dependency, and the blind spot is the permanent consequence. The octopus eye evolved from a different ancestral structure, in which the photoreceptors happen to be oriented the right way from the start. No correction needed. No blind spot produced. The same functional goal, a camera-style image-forming eye, achieved in two different ways, from two different starting points, with one of those ways producing a minor but real improvement over the other. Evolution converged on the same design twice and got it slightly wrong the first time. The octopus also perceives light in a dimension that is entirely invisible to human eyes without instruments, polarized light. This is worth explaining because it is easy to assume that polarization is an esoteric or specialized property of light that rarely matters in natural environments. It is not. Polarized light is everywhere in aquatic environments. When light enters the water from above and scatters off particles in the water column, off suspended organisms, off the surfaces of fish and other animals, it becomes partially polarized in ways that vary with direction, depth, and the specific properties of the scattering object. The reflective surfaces of fish scales are strongly polarizing, producing a characteristic polarization signature that differs from the background in ways that a polarization-sensitive eye could potentially use to detect fish against a scattered light background. Underwater surfaces, including the surfaces of other animals, often have polarization properties that carry information about their composition and orientation that intensity-based vision cannot access. The octopus photoreceptors have a physical orientation that makes them sensitive to the direction of polarization of incoming light. This gives the octopus an additional dimension of visual information, not just how bright and, possibly, what color, but also how the light is polarized, which adds information about the surface properties and orientation of the objects being viewed. One hypothesis that has attracted research attention is the possibility that the iridescent skin patterns of cephalopods are not just visual displays in the conventional sense, but polarization signals, patterns that carry information specifically in the polarization dimension that is visible to other cephalopods but invisible to predators whose vision, like ours, is polarization blind. A cuttlefish displaying a pattern that looks uniform to a human observer might be displaying a complex polarization pattern visible to another cuttlefish nearby. The exchange of information could be happening in plain sight of any number of observers, and only the recipients, the other cephalopods with the right visual equipment, would receive the message. Whether something similar is true of octopuses remains actively debated, but the possibility is real. The optical evidence is consistent with it, and the behavioral evidence, while not yet conclusive, does not rule it out. There is one more aspect of the octopus's sensory relationship with light that deserves extended attention, because it is one of the more recent and more surprising discoveries about the animal's biology. The skin of the octopus appears to be sensitive to light independently of the eyes. This was discovered somewhat accidentally, the way many important biological findings are discovered, during genomic and transcriptomic analyses aimed at characterizing gene expression patterns in different tissues. Researchers found that the skin of octopuses expresses opsin genes. Opsins are the protein family that underlies light sensitivity in all animal visual systems. They are the molecules that absorb photons of specific wavelengths and initiate the chain of biochemical events that eventually produces a neural signal. They are the molecules that make eyes work. Finding them expressed in skin cells was not what anyone expected. The skin is not an eye. It does not have the optical components needed to form an image, no lens, no focused light, no spatial arrangement of photoreceptors capable of resolving detail. But it has the molecular machinery to detect the presence of light and, potentially, to respond to it. The most widely discussed hypothesis is that dermal photosensitivity contributes to the camouflage system. Not by providing the visual information that the central brain uses to select camouflage patterns, that information comes from the eyes, through the conventional visual pathway. But perhaps by allowing individual regions of skin to make local adjustments to their color and texture in response to local lighting conditions, without waiting for instructions from the central brain. A patch of skin that can detect whether it is in shadow or in direct light might be able to fine-tune its chromatophore state in ways that improve the match to the immediate local environment, operating simultaneously and in parallel across the entire body surface, at a resolution that no central control system could efficiently manage. If this is true, and the evidence is suggestive but not conclusive, then the octopus does not merely see with its eyes. It perceives light, in some form, with its entire body. Every square centimeter of skin is, in some sense, a light-sensitive surface. The resulting perceptual experience, if the octopus has perceptual experience at all, would be structured around a relationship with light that is qualitatively different from anything we can construct from our own two forward-facing eyes. It would be something more distributed, more enveloping, more like being continuously bathed in a medium that carries information about the world on every surface of the body simultaneously, with the eyes serving as the regions of highest resolution and most sophisticated processing in a much larger network of photosensitivity that extends in every direction. Not seeing, exactly, in the way we use that word. Something for which we do not have an adequate word at all. All octopuses are venomous. Every single species in the group, and there are more than 300 of them, distributed across every ocean on Earth from coral reefs to abyssal plains, produces venom in specialized salivary glands and uses that venom as part of the process of predation. This is one of the most consistently surprising facts about the octopus to people who encounter it without prior knowledge, because nothing about the animal's image in popular culture prepares you for it. Cobras are venomous. Scorpions are venomous. Box jellyfish are venomous. These are animals whose dangerous qualities are woven into the mental category they occupy, part of what you think of when you think of them. The octopus does not occupy that mental category. It seems soft, curious, intelligent, a little alien but not threatening. And yet every one of them carries venom. The venom system consists of two distinct salivary glands, each producing a different component. The posterior gland produces digestive enzymes, proteases, lipases, the biochemical tools needed to begin breaking down prey tissue into absorbable nutrients. These are not venom in the sense of toxic compounds that harm other organisms. They are the beginning of the digestive process, delivered externally before the prey is consumed. The anterior gland produces the toxins, properly speaking, the compounds that interfere with the prey's nervous system, paralyze its musculature, and prevent it from escaping or resisting while the octopus feeds. The specific toxic compounds vary between species, but typically include proteins that block neural transmission, compounds that cause muscle relaxation, and in some cases compounds that act on multiple physiological systems simultaneously. For the prey items that octopuses normally hunt, crustaceans, mollusks, small fish, these compounds are highly effective, producing rapid incapacitation that allows the octopus to handle the prey safely and consume it without the risk of injury from struggling. For humans, the venom of most octopus species produces effects ranging from no perceptible effect at all to localized pain and swelling at the bite site, usually resolving within hours to days without treatment. The beak is small, the venom dose delivered to a human is generally low relative to body mass, and the specific toxic compounds in most species are not well optimized for the physiology of large vertebrate mammals. Most of the time, being bitten by an octopus is a minor and temporary inconvenience. There is one exception. One species complex that departs from this pattern so dramatically that it has become one of the most discussed examples of extreme biological danger in a small package. The blue-ringed octopus, belonging to the genus Hapaloclina, comprising 4 recognized species distributed across the Indo-Pacific from Australia to Japan, is one of the most venomous animals on the planet, gram for gram. The animal is small, very small. The largest individuals reach about 20 centimeters in total span with arms extended, and most are considerably smaller. It fits comfortably in the palm of a hand, and in terms of weight it is measured in tens of grams, not kilograms. Its size places it firmly in the category of animals that most people would pick up without a second thought if they found one in a tide pool or on a beach. Its coloration is beautiful in the way that biological warning signals often are, a background of yellow ochre or pale brown, covered in vivid blue rings with darker edges that pulse visibly when the animal is disturbed or agitated, the rings flashing repeatedly from pale to brilliant blue in a display that has the quality of urgency about it to any observer with the background to interpret it. This pulsing is an aposematic signal, a visual advertisement of danger, evolved to communicate to visually capable predators that the animal is not worth attacking. It is the visual equivalent of a rattlesnake's rattle, an honest warning that the consequences of proceeding will be unpleasant. For predators that have learned the signal, it works. For humans who have not, the pulsing blue rings, often read as fascinating rather than dangerous, the kind of display that makes you want to look more closely, to pick it up and examine it, to show it to the people you are with. And that is where the danger lies. The primary toxin in the blue-ringed octopus venom is tetrodotoxin, a compound that, by the standards of biological chemistry, is extraordinarily potent. Tetrodotoxin works by blocking the voltage-gated sodium channels in neurons, the molecular machines that neurons use to generate and propagate electrical signals. When these channels are blocked, neurons cannot fire. They become electrically silent. The signals that would normally travel from the brain to the muscles, instructing them to contract, cannot propagate. The muscles that control breathing cannot receive the instruction to breathe. The diaphragm stops. Respiratory arrest follows, and unless the victim receives immediate artificial ventilation, death from hypoxia follows the respiratory arrest within minutes. There is no antidote to tetrodotoxin. The treatment for severe envenomation is entirely supportive, keeping the victim alive through mechanical ventilation while the body's metabolic machinery slowly breaks down and eliminates the toxin, a process that can take several hours. The victim may remain fully conscious throughout. Tetrodotoxin does not affect consciousness at the doses produced by a blue-ringed octopus bite, and may be fully aware of the paralysis, unable to breathe, unable to move, able to feel and hear and understand what is happening but completely unable to communicate or to act. A single blue-ringed octopus carries enough tetrodotoxin to kill, depending on the species and the individual, somewhere between 10 and 26 adult humans. This estimate is based on the measured tetrodotoxin content of venom glands from specimens collected for research, compared against the known lethal dose for vertebrates. The number is startling in the context of an animal that weighs less than 30 grams. The toxicity, scaled to body mass, has very few equivalents anywhere in the animal kingdom. What makes the biology of this system particularly interesting is the source of the toxin. The blue-ringed octopus does not synthesize tetrodotoxin itself. Its own metabolic machinery is not capable of producing the compound. The tetrodotoxin is produced by bacteria, symbiotic bacteria belonging to several genera, including Vibrio and Pseudomonas, that live within the tissues of the octopus's salivary glands. The bacteria produce the toxin as part of their own biochemistry. The octopus provides the bacteria with a protected, nutrient-rich environment in which to live. And the octopus acquires, through this symbiotic relationship, a chemical weapon of extraordinary potency that it could never have produced on its own. This kind of toxin-producing symbiosis appears in multiple, unrelated lineages of venomous animals across the animal kingdom, in poison dart frogs, in pufferfish, in several species of newts, in some marine invertebrates. The pattern is consistent enough to suggest that it represents a generally accessible evolutionary strategy, that acquiring toxic bacteria as symbionts, and directing their toxin production toward the animal's own defensive or predatory purposes, is a path that natural selection finds and exploits repeatedly because it offers enormous benefits at relatively low metabolic cost to the host. The hunting behavior of the octopus is one of the most behaviorally rich aspects of the animal's ecology, and it repays careful attention because it reveals, in practical terms, what the cognitive machinery of the nervous system is actually doing day to day. The octopus is, at its core, an ambush predator. Its preferred strategy is patience, settling into camouflage, becoming invisible to the environment, and waiting for prey to come within reach before launching a rapid strike. This is a strategy that makes particular sense for an animal whose jet propulsion is effective over short distances but energetically expensive to sustain, and whose best physical attribute is not speed across open water but the remarkable reach and grip of its arms. Waiting burns very little energy. Waiting, done correctly, is nearly invisible. And when prey comes close enough, the strike takes fractions of a second, too fast for most prey animals to respond effectively. But the octopus is not rigidly committed to any single hunting mode. It hunts actively as well as passively, moving through reef structures and exploring crevices, lifting rocks and shells to investigate what might be hiding beneath them, systematically covering territory in a way that reflects genuine spatial memory of what has been checked and what has not. Some individuals develop specific hunting territories, learning the layout of a reef section well enough to exploit it efficiently and returning to productive areas based on past experience. For fish prey, the timing and precision of the attack is critical. High-speed video analysis shows that the extension of the arm toward a fish target takes somewhere between 30 and 100 milliseconds, a timeframe that is shorter than the reaction time of most fish to visual stimuli, and shorter than the time needed for a fish to initiate an escape response. The octopus is faster than the fish's ability to respond, which means the fish has no opportunity to flee once the attack has begun. The suckers activate along the contact surface almost simultaneously, creating adhesion that the fish's muscles typically cannot overcome. The beak follows immediately, delivering venom to initiate paralysis and injecting salivary enzymes to begin digestion. For shelled prey, bivalves, crabs, sea urchins, the situation is completely different and considerably more interesting from a behavioral standpoint. A clam sealed in its shell is not accessible through the direct application of force to soft tissue. The octopus must either find a way to open the shell, create an entry point by drilling through it, or chemically persuade the mollusk to relax the muscles that keep it shut. All three strategies are used, often in sequence, by the same animal on the same prey item. The opening strategy involves applying torsional force across the shell, gripping with suckers on both valves and twisting in opposite directions, or pulling in opposite directions while a beak-shaped wedge is inserted into the gap. The force required is substantial, and different prey species resist with different strengths. An octopus that has successfully opened a specific species of bivalve before approaches new individuals of that species with the technique refined by experience, applying force at the specific angle and leverage point that has proven effective, spending less time exploring alternatives than a naive animal would. When direct opening fails, drilling begins. The beak carves through the shell at a location chosen with what appears to be deliberate care, near the adductor muscle, where its chemical relaxation will be most useful, at a thinner region of the shell, where less work is required. Through this hole, saliva containing both neurotoxin and digestive enzymes is injected. The neurotoxin paralyzes the adductor muscle, causing the shell to relax and open. The digestive enzymes begin breaking down the tissue inside. After several minutes, the octopus waits with patience. During this stage, the contents can be drawn out through the same hole in a partially liquefied state, combining predation and digestion in a single economical process. The behavioral sophistication of this feeding sequence, with its multiple strategies deployed in context-sensitive sequence, its use of chemical tools, its refinement through experience, and its adjustment to the specific properties of different prey species, is one of the clearest demonstrations in natural behavior of a cognitive system doing something more than executing fixed motor programs. Fixed motor programs do not learn. Fixed motor programs do not try one approach and then switch to another when the first fails. Fixed motor programs do not choose where to drill based on knowledge of the prey's anatomy. What the octopus does when it hunts shelled prey looks, in its structure, very much like problem solving. The problem is presented: there is food inside this shell, how do I get to it? Multiple approaches are available. Some work better than others for specific prey items. Experience improves performance. The solution is found, or not found, through a process that is genuinely exploratory rather than prescripted. The relationship between the octopus and its habitat is another expression of the same cognitive sophistication. Octopuses establish dens, home bases, shelters in which they rest, store prey remains, and retreat when threatened. The selection, construction, and maintenance of a den involves behavioral choices that go well beyond simply occupying the most convenient available cavity. Octopuses choose dens with specific geometric properties, entrances narrow enough to be defended with a few arms while allowing rapid exit, interior volumes large enough to accommodate the body, positions with good sight lines to the surrounding area. When suitable natural cavities are not available, they create them, digging into soft sediment, excavating sand from beneath rocks to create chambers, stacking stones and shells to build partial walls that define a sheltered space. The entrance of an established octopus den is often marked by an accumulation of prey remains, empty shells, crab carapaces, the external elements of prey that the octopus did not consume and simply discarded near the entrance after eating. This accumulation, called a midden by researchers who study it, is not merely debris. It is evidence of the systematic hunting activity of a specific individual, building up over days or weeks of occupation. The composition of the midden reflects the prey available in the surrounding territory and the individual preferences of the occupant, which vary between individuals in ways that have been documented in multiple species. Some octopuses have been observed decorating the entrance of the den with specific objects that appear to serve defensive or concealing functions, positioning shells to partially block the opening, arranging stones in configurations that narrow the entrance, placing visually disruptive objects at positions that might draw a predator's eye away from the actual opening. Whether these arrangements are deliberately strategic, whether the octopus is actually reasoning about how its den entrance looks from outside and choosing decorations accordingly, or whether they reflect simpler behavioral tendencies that produce adaptive outcomes without conscious strategic intent, is a question that behavioral researchers engage with carefully, because it is exactly the kind of question where the line between interpretation and anthropomorphism is easy to cross without noticing. What is clear is that the behaviors are real, are consistent within individuals, and are not adequately explained by reflexive responses to immediate stimuli alone. An octopus placing a shell at a specific position in a den entrance is doing something that is motivated by properties of the overall arrangement, not just responding to the shell's immediate presence, but doing something about its position relative to other elements of the structure. That requires a representation of the structure as a whole, not just the element being manipulated. One of the most discussed examples of octopus tool use, a behavior that was considered sufficiently remarkable when documented to warrant publication in a major scientific journal, involves not den construction but portable shelter. The veined octopus, found in the coastal waters of the Indo-Pacific, has been repeatedly observed collecting discarded coconut shell halves from the seafloor, carrying them to new locations, and assembling them into portable shelters that the animal can retreat into when threatened. The shells are carried under the body, gripped by the suckers, while the octopus walks on its remaining arms in an awkward stilt-like gait that is much slower and more conspicuous than normal movement. The carrying is the remarkable part. The octopus is not immediately using the shell for shelter when it picks it up. It picks it up, accepts the cost of the awkward locomotion it imposes, carries it some distance, and then uses it later. This temporal separation between the acquisition of the tool and its use, carrying a thing in anticipation of a future need, is what makes behavioral researchers call it planning, or at minimum prospective cognition. The octopus is doing something now because of something it expects to need later. Whether that constitutes planning in the full cognitive sense or something more automatic is debated. But the behavior itself is documented, replicated, and real. The intelligence of the octopus, as a scientific subject, is methodologically challenging in a way that is worth understanding before reviewing the evidence, because the methodological challenges are themselves informative about the nature of the animal's cognition. Most of what we know about animal cognition comes from experiments designed for vertebrates, tasks that present choices between visual stimuli, that reward specific behavioral responses, that use food as a motivator, that are implemented in laboratory spaces designed around the physical capabilities and sensory systems of mammals and birds. These methods are well validated, highly reproducible, and have produced an enormous body of reliable knowledge about the cognitive abilities of the animals they were designed for. They do not work well for octopuses. The reasons are multiple and instructive. Visual discrimination tasks designed for vertebrates often involve stimuli presented in specific visual angles and formats that vertebrate eyes naturally engage with, but octopus eyes are positioned differently, process information differently, and the animal may not be inclined to look at a stimulus panel the way a rat or a pigeon would. Tasks that reward food are complicated by the octopus's feeding biology, which involves complex sensory evaluation of prey before consumption. The animal may need to taste a reward item before deciding whether to eat it, which can confound the logic of a task designed to measure whether the animal chose the rewarded option. Apparatus designed for animals with paws or beaks may be physically awkward for an animal with 8 sucked arms that can approach from any direction. Perhaps most importantly, the octopus is easily bored. An animal with high curiosity and sophisticated exploratory behavior will quickly habituate to a laboratory task that is designed to be repeated many times for reliable statistical estimates. After a few repetitions, an octopus may begin to interact with the apparatus in ways the experimenter did not intend, investigating the supports, attempting to dismantle the test chambers, squirting water at the experimenters. These behaviors are classified as failures or excluded from analysis. But they may be evidence of a cognitive style that is not well served by the repetitive, structured format of standard cognitive testing. When tests are redesigned to accommodate octopus biology and behavior, presented in formats that match the animal's sensory system and behavioral tendencies, offered in sessions short enough to maintain motivation, implemented in contexts that make ecological sense, the results are consistently and sometimes dramatically impressive. One of the early experiments that substantially shifted the field's understanding of cephalopod cognition was conducted in the 1990s by Jennifer Mather and Roland Anderson, and it involved something apparently simple. Octopuses were given sealed plastic bottles with food inside. The task required the animal to identify that food was inside, understand that the container needed to be opened to access it, and discover how to open it through trial and error. The finding was not just that octopuses could solve the problem, which was already interesting enough. It was that different individual octopuses developed different solutions. Some wrapped arms around the bottle and applied continuous rotational torque until the cap loosened. Others inserted the beak into the gap between cap and bottle and applied leverage. Others held the bottle with several arms and pulled in opposing directions on the cap with others. These solutions were idiosyncratic, distinctive to specific individuals, retained and refined across repeated trials, not abandoned when they worked in favor of trying something new. The researchers named what they were observing individual problem-solving styles. The broader concept that their work contributed to is what is now called octopus personality, the existence of stable individual differences in behavior that persist across time, across contexts, and across the life of the animal. Not all octopuses approach novelty the same way. Not all octopuses are equally exploratory or equally cautious or equally persistent in the face of difficulty. These differences are real, are measurable, and are not reducible to differences in health or age or immediate motivational state. They are traits. Stable behavioral traits, which is what personality means in the precise sense. Memory in the octopus is more robust and more sophisticated than its short life would suggest if you assumed that memory capacity scales with lifespan. It does not, at least not in the octopus. Classical conditioning experiments, in which a neutral stimulus is consistently paired with a meaningful one until the animal responds to the neutral stimulus as if it predicts the meaningful one, show that octopuses acquire associations after only a few pairings, often fewer than the number required for the same task in vertebrates. They retain these associations for weeks to months, a significant fraction of the animal's total lifespan. They generalize the associations to new stimuli with appropriate similarities, applying what they have learned in one context to analogous contexts they have not encountered before. But perhaps more interesting than the raw measures of acquisition and retention is the qualitative character of what the octopus remembers. There is experimental evidence suggesting that the memory formed in these situations is not merely associative, not merely a link between a stimulus and a response built up through repetition, but something more structured and more contextually specific. In experiments where octopuses were fed in different locations at different times of day, where food was available in location A in the morning and location B in the afternoon, for example, the animals did not simply learn that both locations were associated with food. They learned which location was productive at which time. They adjusted their behavior in anticipation of the schedule, moving toward location A in the morning before feeding began and toward location B in the afternoon, predicting where food would be based on when it was, integrating temporal information with spatial information in a way that required both to be retained and combined. This integration of when and where into a unified memory is called episodic-like memory by researchers, because it resembles the episodic memory that humans use to remember specific past events in their spatial and temporal context. The ability to remember not just that something happened but when it happened and where it happened is considered one of the more sophisticated forms of memory, and its presence in the octopus was not expected on the basis of what was known about invertebrate cognition before these experiments were conducted. The neural substrate of this memory has been partially identified. The vertical lobe of the octopus brain, a structure in the dorsal region of the central brain mass that has no obvious homologue in vertebrate neuroanatomy, is essential for long-term memory consolidation. Octopuses with experimental lesions to the vertical lobe learn normally in short-term memory tasks. They can hold information for minutes and use it within the trial. But they fail consistently to retain what they learn across sessions. Information learned one day is not available the next. The long-term consolidation that would normally occur during or after learning simply does not happen without the vertical lobe. This dissociation between short-term and long-term memory, implemented in distinct neural structures with different functions, is a pattern that appears in vertebrate neuroscience as one of the most reproducible and theoretically significant findings about how memory works. Its presence in the octopus, emerging from completely independent evolutionary pressures in a nervous system built on completely different principles, is another entry in the growing catalogue of cognitive convergences between the octopus and the vertebrates. Observational learning, the capacity to acquire new knowledge or skills by watching other individuals, without any direct trial and error experience of one's own, was for many years considered to be a capacity essentially restricted to social species. The reasoning was straightforward and seemed intuitively compelling. Learning by observation requires the presence of others performing the behaviors to be observed. Social animals, by definition, have regular access to such others. Solitary animals do not. Therefore, selection pressure for observational learning should be weak or absent in solitary species, and the capacity should not develop. The octopus is solitary in the strongest possible sense. It does not live in groups. It does not have persistent social relationships. It generally avoids contact with other octopuses outside of the brief reproductive period, and even then the interaction is typically cautious and often aggressive. The expectation, based on the standard theoretical framework, was clear. The expectation was wrong. Experiments in which octopus observers were allowed to watch octopus demonstrators performing a discrimination task through a transparent partition found that observers subsequently performed the same task significantly faster and more accurately than naive control animals that had not observed a demonstrator. The effect was robust, not a marginal statistical trend, but a clear and consistent performance advantage that was replicated across multiple studies. In some experiments, observer octopuses performed the task correctly on their very first attempt, apparently having extracted the critical information, which stimulus to choose, entirely from observation, without any personal trial and error experience. This result required some revision of the theoretical framework about the relationship between sociality and observational learning. The capacity exists in the octopus despite the solitary lifestyle, which means that the evolutionary pressure that maintained it must have operated in a context other than intraspecific social learning. One plausible alternative: an animal that spends its life carefully watching its environment for predators and prey, that survives largely by accurately reading the behavior of other animals, might develop a general capacity to extract information from observing others that extends to conspecifics as a side effect. An octopus that can watch a fish respond to a visual stimulus and infer something about where food is likely to be, or watch a crab flee in a specific direction and infer where a predator approached from, has a general observational learning capacity that simply happens to work when the observer is another octopus. The way an octopus arm extends to reach a distant target was studied in detail by researchers working at the intersection of biology and engineering, and what they found was both counterintuitive and immediately useful to people designing robotic systems. Extending a highly flexible arm with effectively infinite degrees of freedom to reach a specific point in space is, mathematically, a problem of great complexity. If you tried to solve it the way a conventional robotic controller would, by computing the optimal configuration of every point along the arm's length, then sending instructions to each segment, then coordinating the execution, the computational load would be enormous, and the solution would be fragile to perturbations in the environment or errors in the initial computation. The octopus does not solve it that way. When an octopus reaches toward a target, the arm generates a wave of stiffness that propagates from the base toward the tip. At the leading edge of this wave, the arm is undergoing active bending, creating what functions temporarily as a joint, a localized pivot point. Behind the wave, the arm has already found its final position and is stiffening to hold it. Ahead of the wave, the arm is still in its initial relaxed state. The wave moves the effective joint point progressively along the arm's length, through the geometry of the reach, until the tip arrives at the target. This solution eliminates the coordination problem. You do not need to simultaneously plan the position of every point along the arm. You only need to control a single propagating wave, adjusting its speed and the degree of bending at the wave front to aim the tip at the target. The computation is local and sequential rather than global and simultaneous. The arm essentially solves its own kinematics, using a simple rule that produces the correct result without requiring the nervous system to explicitly compute what that result should look like at every intermediate stage. When engineers working in soft robotics learned about this mechanism, they recognized it immediately as a solution to a problem they had been struggling with. Soft robotic arms with wave-based control have been built and demonstrated, and they do exhibit the flexibility and adaptability advantages predicted by the biological model. The octopus, across millions of years of selection pressure from the environment, discovered a control algorithm that human engineers would have been proud to design. The reproduction of the octopus, and the life cycle that frames it, is marked by a finality that is genuinely striking when you consider what the animal is capable of. A creature that can learn, that can remember, that can recognize individuals, that can solve novel problems, that can use tools and plan for the future, lives for roughly a year to 2 years, reproduces once, and dies. The entire arc of a complex and capable life, compressed into a window that most vertebrates with comparable cognitive abilities would spend entirely in juvenile development. For females, the reproductive period is the last thing they do. After mating, the female finds or constructs a den in a well-protected location and deposits her eggs, long strings of small, pearlescent capsules attached to the substrate or to the den structure in carefully arranged clusters. A female may lay between a few dozen and several 100,000 eggs, depending on species, each one a tiny self-contained developmental system carrying enough nutrients for the embryo to develop to completion without any additional resources from the environment. What follows is the most intensive period of the female's life, and also the last. She does not leave the den. She does not eat. She spends the entire incubation period, which can last weeks to months depending on species and temperature, aerating the eggs by moving water across them continuously with her arms, a gentle, rhythmic fanning motion that ensures adequate oxygen reaches the developing embryos. She removes parasites and debris. She defends the den against anything that approaches, with a ferocity that octopuses in other contexts rarely exhibit. She tends the eggs with a consistency and dedication that, if we observed it in a vertebrate, we would not hesitate to call maternal. And she deteriorates progressively throughout. The proteins of her own muscles are catabolized to provide the energy she needs to keep functioning during the fasting period. The quality of her camouflage declines as the neural systems that control it begin to fail. Her responses become slower. Her movements become less coordinated. She continues tending the eggs through all of this, even as the animal that is tending them becomes less and less capable of doing much else. The longest documented case of this behavior was recorded by researchers who monitored a female of a deep-sea octopus species on the floor of the Pacific Ocean, returning to the site repeatedly over a period of time and finding her still at her post. The total duration of the brooding period they documented was 53 months. More than 4 years. More than twice the normal maximum lifespan of most shallow water octopus species. The female was still alive at the end of it, barely, and the eggs hatched. She was found dead shortly after. This is not the consequence of exhaustion in any passive sense. It is a programmed process, actively controlled by the octopus's own endocrine system. The optic glands, paired structures located between the eyes and the central brain, release hormonal signals after reproduction that trigger the senescence process, the systematic deterioration that ensures the female continues investing in the eggs until they hatch, at the cost of her own continued existence. Researchers who surgically removed the optic glands from females immediately after egg laying found that those animals abandoned the eggs, resumed normal feeding behavior, and lived significantly longer than intact females. The death is chosen by the biology, written into the program, activated by the same glands that triggered reproduction in the first place. For males, the process is similar in its outcome if different in its timing. After mating, males enter a senescence that progresses regardless of environmental conditions or nutritional state. They stop eating, lose their camouflage, become progressively less responsive to the environment, and die within weeks to months. The optic glands, here too, appear to control the process. The evolutionary logic of programmed senescence after reproduction is not difficult to construct in outline, though the details remain debated. An octopus that survives after producing offspring competes with those offspring for the same resources, the same prey, the same shelter, the same territory. If the offspring benefit more from the resources than the parent would, if the parent's marginal contribution to the next generation's survival is greater through dying than through continuing to compete for resources, then selection will favor the program that terminates the parent's life after the reproductive contribution is complete. The genes in the offspring are the same genes that were in the parent. From the gene's perspective, what matters is not which body carries it forward, but how many copies of it survive. The discovery that octopuses sleep was not immediate, because octopuses do not close their eyes. There are no eyelids, the eye is always open, always apparently directed at the world. Recognizing sleep in an animal without the visual cue of closed eyes required behavioral observation that was careful and systematic, looking for the other behavioral signs that sleep produces in animals that are known to sleep. What researchers found is that octopuses show periods of behavioral quiescence that satisfy every operational criterion used to define sleep in zoological research. Relative immobility with a characteristic posture, arms gathered beneath the mantle, body settled against a surface, the overall configuration relaxed rather than alert. Reduced responsiveness to external stimuli that would provoke an immediate reaction in an alert octopus produce delayed or no reaction during these quiescent periods. A characteristic skin state, pale, relatively uniform coloration, without the dynamic patterning of active animals. Rapid reversibility with sufficiently intense stimulation, the animal can be awakened, distinguishing true sleep from illness or incapacitation. And homeostatic pressure, the property that distinguishes sleep from simple inactivity. When octopuses were prevented from sleeping by gentle disturbances, they showed increased sleep pressure in subsequent periods, sleeping more and more deeply when the disturbance was removed. This rebound of sleep after deprivation is one of the most reliable indicators that sleep is a genuine physiological state with real functional requirements, not simply a behavioral choice that can be indefinitely deferred. The effects of sleep deprivation in octopuses parallel the effects seen in sleep-deprived vertebrates in qualitative terms. Reduced responsiveness. Lower accuracy in hunting. Impaired coordination. Compensatory increases in sleep duration when the opportunity arises. Whatever sleep does for the octopus nervous system, the nervous system needs it done regularly, and the consequences of not doing it are real and measurable. Within the sleep periods, two distinct phases were identified. The first phase is quiet, characterized by deep immobility, pale uniform skin, slow regular ventilation of the mantle, a physiological state that looks externally like the deep, restorative sleep that vertebrates spend most of their sleeping time in. The second phase is dramatically different. During the active phase, the sleeping octopus undergoes rapid, complex changes in skin color and texture. Patterns appear and dissolve across the mantle. Waves of chromatic activity move across the skin. The arms twitch and shift. The eyes move beneath closed, not closed. The eyes are always open, but beneath inactive surfaces that are no longer tracking the environment. The ventilation rhythm changes. The whole animal, moments before still and pale, is now restless and vivid in a way that looks almost like wakefulness, and yet the animal is clearly not awake. It does not respond to stimuli at normal waking thresholds, and its overall behavioral state is that of sleep. This active phase, with its patterned skin activity and motor twitching, drew immediate comparison to REM sleep when it was first systematically documented. REM sleep in vertebrates is associated with rapid eye movements and motor twitches, with high-frequency brain activity resembling the waking state, and in humans and other animals with memories robust enough to report on subjectively, with vivid dreaming. The consolidation of episodic and emotional memories appears to happen partly during REM sleep. The reactivation of neural patterns laid down during waking experience, essentially a rehearsal or replay of the day's events in compressed and reorganized form, has been measured directly in the sleeping brains of rodents and other vertebrates. The octopus does not have the same neural circuits that generate REM in vertebrate brains. The structures and mechanisms are different, but the phenomenology of the active phase, its cyclic occurrence, its distinctive character compared to the quieter phase, and especially the visual character of the skin patterning during it, are consistent with a process that resembles, in its functional logic, what REM sleep appears to do in vertebrates. Something is being processed. Something related to visual experience, if the skin patterns are any guide. Something that happens in regular cycles throughout the sleep period and that the nervous system needs to do. The skin patterning during this phase is what captured the public imagination when video footage of sleeping octopuses became widely available. An octopus in the active phase of sleep can be seen cycling through sequences of skin patterns that resemble, sometimes remarkably closely, the patterns the animal produces when awake and interacting with the environment. Camouflage patterns appropriate to specific substrates. Hunting coloration. Defensive displays. The patterns cycle through at an accelerated pace, changing every few seconds, as if a compressed film strip of the animal's recent waking experiences is being played back through the skin. Whether this represents literal memory replay, the neural patterns of specific waking experiences being reactivated during sleep, as happens in rodent hippocampi during slow-wave sleep, is not yet established. The skin patterns could be the output of such replay, visible through the animal's unique property of having its emotional and experimental state manifested externally in its skin. Or they could be something else, spontaneous activity in the pattern-generating circuits of the skin control system, structured by the regularities of recent experience without constituting literal replay. The distinction matters for understanding what is happening functionally. It is not yet possible to make with certainty. What is certain is that something significant happens during these active phases. The nervous system is doing something that requires the kind of neural activity associated with active processing, not just maintenance, not just cellular housekeeping, but something that engages the systems responsible for perception and behavior in a way that manifests externally in the most expressive display surface the octopus has. And the question of what that something is, and whether it involves anything like the experience we call dreaming, is one that the neuroscience of cephalopods is only beginning to develop the tools to approach. In 2012, a group of some of the most prominent neuroscientists in the world gathered in Cambridge and signed a declaration about the nature of consciousness in non-human animals. The Cambridge Declaration on Consciousness affirmed that the scientific evidence available at that time indicated that non-human animals, including all mammals, all birds, and a number of other creatures including octopuses and other cephalopods possess the neurological substrates that generate conscious states. The declaration was careful in its language. It did not claim that these animals are conscious in the same sense that humans are conscious, or that their subjective experience, if they have any, resembles ours in content or richness. It claimed something more limited but still significant, that their nervous systems are complex enough, and the neurological correlates of consciousness identified in vertebrates are sufficiently present in them, that the possibility of conscious experience cannot be dismissed a priori. That the question of whether they experience anything deserves to be treated as a genuine scientific question rather than as something to be assumed away. For the octopus specifically, this question is more philosophically tangled than for any other animal on the list. Not because there is less evidence of cognitive sophistication, if anything, the evidence for sophisticated cognitive processes in the octopus is as strong as or stronger than the evidence for most other invertebrates. But because the architecture of the octopus nervous system is so different from the architecture for which our theories of consciousness were developed. Our theories of consciousness, the ones that identify specific neural correlates of conscious experience, that link awareness to particular kinds of information integration or global broadcasting or recursive self-modeling, were developed by studying vertebrate brains, mostly mammalian brains, and mostly human brains. They identify specific neural structures and dynamics as the substrate of conscious experience. When those structures and dynamics are present in other animals, we have grounds for hypothesizing that those animals may be conscious. When they are absent, we have grounds for skepticism. The octopus presents a system in which some of those structures and dynamics are present in forms that appear analogous, and others are absent or replaced by something different. The vertical lobe's role in memory consolidation resembles the hippocampus's role in vertebrates. The chromatophore system provides a window into internal state that has no vertebrate equivalent. The distributed processing in the arms creates a situation where there may be multiple semi-independent processing streams that are only partially integrated, which fits awkwardly into theories of consciousness that emphasize unified global integration. The philosopher Peter Godfrey Smith, who has spent years both studying cephalopod neuroscience academically and diving with octopuses in their natural habitats, suggests that if the octopus has subjective experience, it is likely structured differently from ours not only in its content but in its fundamental form. Not a unified stream of awareness anchored in a central brain, but something more fragmented, more distributed, more like a collection of semi-independent experiences that are loosely coordinated by the central brain without being fully unified into a single subject. Each arm experiencing its environment, in whatever sense experiencing applies, with the central brain receiving reports but not fully integrating everything into a single coherent perspective. This is speculative, and Godfrey Smith is careful to present it as such. But it is speculative in a productive direction, because it forces us to ask what we mean when we say that something is conscious, and to notice that our usual assumptions about the unity and centrality of consciousness are assumptions derived from the specific architecture of the vertebrate nervous system, not universal features of what experience must be. The octopus challenges us to imagine a form of awareness, if there is any awareness at all, that is not organized around a single subject in a single brain. A form of awareness that might be multiple, that might be distributed, that might feel, in whatever way it feels, less like a single perspective and more like a collection of perspectives in loose communication. Whether this is possible, and what it would be like if it were, is one of the more genuinely open and genuinely important questions that the octopus puts before us. The octopus's biology has touched the life of medicine and materials science and robotics in ways that are worth surveying briefly before we arrive at the end of tonight's journey, because they illustrate something important about why the animal matters beyond its intrinsic interest as a natural phenomenon. The tetrodotoxin of the blue-ringed octopus, terrifying in the context of an encounter on a beach, becomes something quite different in the context of pharmaceutical chemistry. Tetrodotoxin is an extraordinarily precise and potent blocker of specific voltage-gated sodium channels in neurons, the kind of precision that chemists trying to design pain-blocking drugs dream about. Current local anesthetics and many pain medications work by blocking sodium channels, but they do so with less specificity than tetrodotoxin, affecting channels in multiple tissues and producing side effects that limit their therapeutic use. Tetrodotoxin itself, at the doses that would make it useful as a painkiller, is simply too dangerous for most clinical applications. The margin between an effective analgesic dose and a lethal dose is uncomfortably narrow. But understanding exactly how it works, the precise geometry of its binding to sodium channels, the specific features of the molecule that confer its extraordinary potency and selectivity, is informing the design of analogues that might retain some of the selectivity without the extreme toxicity. Clinical trials have already been conducted, in Canada and several European countries, testing purified tetrodotoxin in low doses for the management of pain associated with advanced cancer, where the risk-benefit calculation is different from normal clinical contexts. The results have been sufficiently promising to justify continued investigation. The skin of the octopus continues to be studied as a design target by materials scientists and engineers who want to build adaptive surfaces. The specific challenge it represents, combining dynamic color change, structural color effects, and texture change in a single surface that responds to environmental information at neural speeds, has not been solved. Each individual component has been approximated in laboratory materials. But integrating all three into a single surface, with the resolution and response speed of the biological system, remains beyond current materials technology. The octopus skin defines the boundary of what is possible, and the boundary is considerably beyond what we have achieved. The soft robotics field continues to use the octopus arm as its primary biological model. Robots with arms made of flexible materials, capable of conforming to complex objects, passing through narrow openings, and operating in environments where rigid structures would fail, have been built in multiple research laboratories around the world. The control algorithms that make these arms useful draw on the principles of distributed control and wave-based reaching that the octopus nervous system implements. The robots are not as capable as the biological system, in any dimension you might measure. But they are considerably more capable than rigid robots in the environments where softness and flexibility matter, and the gap is narrowing. There is a perspective on the octopus that only becomes visible when you hold all of the biology we have discussed tonight in a single view, when you try to see the animal whole rather than one feature at a time. The octopus represents something that the history of biology was slow to fully recognize and that philosophers of mind are still working to accommodate. It represents the existence of intelligence as a general possibility of matter, not as a specific property of a specific architecture. It demonstrates that the cognitive capacities we associate with advanced nervous systems, learning, memory, problem-solving, tool use, play, individual personality, possibly dreaming, possibly consciousness, can emerge from architectures that look nothing like the architecture we discovered them in first. This has implications that extend beyond biology. If intelligence can emerge from distributed processing, from nervous systems that do not centralize computation in a single brain, from bodies whose processing hardware is embedded in the limbs rather than concentrated in the head, then intelligence is less fragile, less contingent, less dependent on a specific and improbable sequence of evolutionary events than it sometimes appears when you look only at vertebrates. The octopus is an existence proof that the capacities we value in ourselves are achievable by a different route. That evolution found two roads to something like a mind, using completely different maps. Every time the field has drawn a line around the capacities that count as genuinely intelligent, the octopus has appeared on the other side of it. Not as a curious exception that leaves the general rule intact, but as a demonstration that the rule was drawn too narrowly, that it captured a sample rather than a category, that the space of possible minds is larger than the sample suggests. The octopus is not a vertebrate that lost its spine. It is not a simplified version of something more complex that we understand better. It is a complete alternative approach to the problem of being a complex, aware, capable organism in a world full of challenges. Built from different materials, organized around different principles, subject to different constraints, arriving at different but overlapping solutions. 500 million years of evolution, working on completely separate raw materials, building something that we can recognize as a kind of mind. That recognition itself is worth pausing on, that across 500 million years of separate history, across every physical and biological difference between a mammal sitting somewhere dry and warm and a soft invertebrate moving through cold seawater, there is still enough in common to produce something that looks, across that enormous distance, like recognition. An octopus in a tank looking at a researcher. A researcher looking back. something happening in the exchange that is not nothing, that is not merely mechanism, that has the quality, however we describe it, however carefully we hedge the description, of two kinds of awareness briefly finding each other. That is, perhaps, the most interesting fact of all. Somewhere in the ocean right now, an octopus is settled into a crevice in a reef, arms gathered inward, skin pale and still. The rhythm of the mantle is slow and regular, the gentle rise and fall of a body at rest. The eyes are open, as they always are, but they are not tracking, not searching, not alert to the movement of prey or the approach of danger. The animal is somewhere else, in whatever sense an octopus is somewhere else during sleep. And at some point in the hours ahead, the skin will begin to change. Patterns will move across it, complex and rapid, colors shifting in sequences that mirror the world the animal navigated during the day. The reef, the sandy patch it hunted across, the crab it spent 20 minutes persuading out of its shell, the large fish it went still for and waited out, something of all of this will pass, somehow, through the skin, visible from outside as a record of a life briefly reviewed. 8 semi-autonomous arms, each containing more neurons than many entire invertebrates, will twitch and shift. The ganglia distributed along their length will fire patterns that were laid down during the day's hunting and exploring and surviving. And the central brain, that small compact structure wrapped around the esophagus in the space between the eyes, will be doing something, exactly what, exactly how, with exactly what relationship to anything like experience, we do not yet know. What we know is that the animal will wake before dawn. It will emerge from the crevice with the unhurried confidence of a creature at home in its environment. It will move through its territory, checking the places where prey tends to accumulate, settling into camouflage when movement in the water suggests something worth watching, adjusting its skin to a sand color and a sand texture with the casual ease of long practice. It will find something to eat, or it will not. It will spend a few minutes with an interesting object it finds wedged between rocks, turning it with its arms in a way that has no immediate practical purpose, and then leave it. It will live its brief and intense and apparently rich life without any awareness of being watched, without any sense that it occupies a central place in anyone's thinking about the nature of mind and the boundaries of consciousness and the question of what forms awareness might take in a universe that has been running biological experiments for 500 million years. And it will do all of this in the ocean, in the dark, in a body that has no bones and 3 hearts and blue blood and 8 partially independent minds, in a life that will last perhaps 1 more year. And the more we look, the more surprising it becomes. And as you drift into a deep, restorative sleep, remember to subscribe, so these stories can continue to find you, night after night. Sleep well, and sweet dreams.